Some of the bacterial affiliates it includes in the classification of bacteria, and they are the prokaryotic. Some are the actinomycetes mycelial eubacteria. Actinomycetes are a group of bacteria that are characterized by their branch multicellular filamentous analogous to the mold. They do not form the endospore, although some of them form conidia on the aerial branch. The conidiophore, because of their branch filamentous structure and the formation of conidia similar to the hyphae and the conidia of fungi, they were once again considered to be fungi. However, it is not well established that they are bacteria and differ from nearly all the true fungi in the following points. The mycelium of actinomycetes is extremely fine and generally is narrower. The cell wall of actinomycetes consists of peptidoglycan, muramic acid, diamphenylic acid, where those of the true fungi are made up of chitin. Actinomycetes shows typical prokaryotic structure, whereas the true fungi shows the eukaryotic. Almost most of the actinomycetes are found in the soil. Some member are the example are Nocardia and streptomycete are responsible for the characteristic smell of the damp soil. A few species of streptomyces are important source of antibiotic. Some of the species are responsible for causing important disease of plant, animal and human being. The common representative groups are Actinomyces, Mycobacterium, Nocardia, Streptomyces, and Micromonospora rickettsii. The rickets are a group of a small pleomorphic obligate intracellular parasite widely distributed among the member of certain blood-sucking arthropods as a flea, lice and ticks. They are not pathogen for the arthropod, but on being transmitted by the arthropod's bite to man, they can cause fatal infection. The rickets possess certain characteristics, for example, extremely small size and the parasitic habit of life, which are found also in virus. Therefore, for many years, they were thought to be closely related to the virus. However, the detailed examination with the electron microscope has revealed that they resemble the bacteria more than that they do to the virus, and therefore they are not now considered to be bacteria. The rickets, for example, multiply by binary fission, contain both DNA and RNA, possess intercellular metabolite enzymes, and are enclosed in a rigid cell wall containing the peptidoglycan. In this feature, they resemble the bacteria. They differ from the virus in their possession of metabolic system. They differ from the bacteria in that all of them are obligate parasite, inability to grow on the inanimate medium. In their persistence to staining with aniline dye and is their intimate association with the arthropod life. They are minute, short rods about zero point 0.2 to zero point 0.5 micron in diameter and zero point 0.3 to 1 micron in length. They are non-motile and do not form endospore. They stain poorly by aniline dye and are gram-negative. They can be stained with a special method of staining. They reproduce by binary fission. This means that they can grow and reproduce only so long as they are present within the living cell of the host plant. At least 7 species have been found in association with the disease in man. Two rickettsial disease of the considerable interest are the typhus fever, Rocky Mountain spotted fever. Typhus fever caused by the Rickettsia provenzeki is transmitted by the body louse from man to man. Rocky Mountain fever is caused by Rickettsia rickettsii and transmitted to the woman by the bites of blood-sucking insect. Chlorotetracycline, chloramphenicol, and the oxytetracycline are the therapeutic values in the treatment of rickettsial disease. Mycobacterium gliding bacteria. The vegetative cell of these bacteria are relatively long, cylinder, flexible, non-flagellated rods. They have no apparent organ of locomotion. They are characterized by a typical gliding motility which occur only in contact with the solid surface. Unlike the eubacteria, they lack a rigid cell wall. Mycobacteria, myco is slime, are commonly called slime bacteria because of the vegetative cell form and multiply within a thin layer of slime producing loose colony. At its maturity, the cell piles up to a certain point and give rise to a structure called the fruiting body. The cells become thickened and develop thick enclosing wall. These spherical refractile resting cells are called the microcyst. Upon germination, they produce new vegetative rods. Thank you.